Anunnaki blood, or
Rh-negative blood refers to a specific classification of blood type based on the presence or absence of a protein called the rhesus factor or Rh factor on the surface of red blood cells. If a person lacks this protein, they are classified as Rh-negative, whereas individuals with the protein are considered Rh-positive. What makes Rh-negative blood unique is the complete absence of the Rh factor. This distinction is rooted in genetics. The absence of the Rh factor is relatively rare, occurring in about 15% of the global population, though its frequency varies significantly among different ethnic groups. Rh-negative blood has important implications for medical science. One key difference lies in the immune system's response. If someone with Rh-negative blood is exposed to Rh-positive blood, their immune system might recognize the Rh protein as foreign and produce antibodies against it. This immune response is known as Rh sensitization. This is particularly significant in pregnancy. If an Rh-negative mother carries an Rh-positive baby, her immune system might attack the baby's red blood cells, causing a condition called hemolytic disease of the newborn, or HDN. To prevent this, medical professionals administer an injection of Rho-D immune globulin, commonly known as Rho-GAM, to Rh-negative mothers during and after pregnancy. The Rh-negative classification also plays a critical role in blood transfusions. People with Rh-negative blood can only safely receive blood from other Rh-negative donors, where Rh-positive individuals can receive blood from either Rh-positive or Rh-negative donors, as long as the ABO compatibility matches. The origins of Rh-negative blood remain a subject of scientific curiosity. While mainstream science attributes it to evolutionary mutations, some speculative theories suggest links to ancient populations or even extraterrestrial ancestry. The O-negative blood, a type of Rh-negative blood, is particularly noteworthy because it is considered universal for red blood cell donations. This means it can be given to almost anyone in need, as it lacks not only the Rh factor but also A and B antigens, reducing the risk of an adverse immune reaction.
That's the basics out of the way. Now let's get down to why we have a blood that we shouldn't have. There are no other known species on Earth with a blood type system exactly like the Rh factor in humans that can lead to immune complications during pregnancy. This leaves us with the question, why would nature do this? It wouldn't. Unless it's either a mutation, or foreign blood bred into us, or added to us in the past to control population.
The idea that Rh-negative blood is the result of a genetic mutation is rooted in mainstream genetics. The Rh factor, specifically the presence or absence of the rhesus D protein on red blood cells, is tied to the Rhd gene. When someone lacks this protein, Rh-negative, the typical explanation is a mutation in the Rhd gene, either deleting it or rendering it inactive. While this explanation aligns with observed genetic patterns, it warrants deeper scrutiny through logic. Mutations are a known mechanism of genetic variation, often caused by copying errors during cell division, environmental factors, or other influences. However, logic demands we ask why this particular mutation would occur, persist, and spread. If Rh-negative blood arose from a mutation, it must have provided some survival advantage or been neutral enough not to disappear due to natural selection. This assumption raises questions. Rh-negative individuals face reproductive challenges when carrying Rh-positive offspring, such as hemolytic disease of the newborn, HDN, which could lead to decreased reproductive success. This seems counterintuitive for a mutation that has persisted for millennia, particularly in isolated populations like the Basques or Berbers, where it is notably prevalent. If we rely on mutation alone, it must explain why Rh-negative blood has not been eliminated or severely diminished due to its reproductive disadvantages. Mainstream genetics often circumvents this by suggesting that the mutation either arose in small populations and persisted due to genetic drift, or had a hidden survival advantage, perhaps linked to disease resistance or environmental factors. While plausible, these explanations often lack direct evidence. For example, no clear disease resistance linked to Rh-negative blood has been conclusively identified, nor has an environmental factor been proven to favor Rh-negative individuals. An alternative perspective challenges the mutation hypothesis. What if Rh-negative blood did not originate as a random mutation, but as a distinct feature introduced into the human genome? This could stem from an interbreeding event with an archaic human species, such as Neanderthals or Denisovans, or even from a separate lineage altogether. The exact timeline of when Rh-negative blood was introduced to humans remains uncertain, but estimates can be made based on genetic studies and logical reasoning. Mainstream science suggests that Rh-negative blood likely originated tens of thousands of years ago, but there is no definitive evidence pinpointing its emergence. Genetic studies indicate that the RhD gene, responsible for producing the Rh protein, underwent a mutation in certain populations. This mutation is often associated with groups like the Basques of Europe, whose distinct genetic traits suggest long-term isolation and ancient origins. Prehistoric times, dies 35,000 to 40,000 years ago, some researchers propose that Rh-negative blood may have appeared during the time of modern Homo sapiens migration and interaction with other hominin species, such as Neanderthals or Denisovans. Interbreeding events during this period could have introduced genetic variations, potentially including Rh-negative blood. However, studies on Neanderthal and Denisovan DNA have not explicitly linked them to the Rh-negative trait, leaving this as speculation. 2. Post-flood era, titled 10,000 to 15,000 years ago. In speculative theories that align with certain ancient texts or alternative histories, Rh-negative blood could have emerged after a significant global event such as the end of the last ice age or the Great Flood. This period corresponds to the rise of advanced civilizations like Mesopotamia, where genetic and cultural developments might have led to unique human traits, including Rh-negative blood.
So where are we? Mainstream have no idea where it came from, no idea how we got it, and no idea why we have it. Let's look now at the possibility of the Anunnaki breeding with Homo sapiens, which created the Homo sapiens sapien, what we call modern man.
90% of the global population has Neanderthal DNA. 5% of the global population, mainly Melanesians and Aboriginal Australians, has Denisovan DNA. 10% of sub-Saharan Africans have trace amounts of Neanderthal DNA via back migration. 100% of sub-Saharan Africans have DNA from unknown archaic humans, e.g., Homo heidelbergensis or others.
The physical fossil evidence for Denisovans is limited. Most of what we know about Denisovans comes from ancient DNA rather than physical remains. It's clear that something created us using Neanderthal, but why didn't we get our H-negative blood then? From the Anunnaki. We didn't because we got the Neanderthal blood. Remember, the Anunnaki genetically created Homo sapiens. It wasn't until 200,000 years later that the Anunnaki bred with the descendants of the first Homo sapiens they created. Mainstream have evidence that Neanderthal and Homo sapiens actually bred together, but what blood type was Neanderthal? Neanderthals likely had the ABO blood group system, similar to modern humans, with evidence pointing to AO, and possibly B blood types depending on their population. Genetic studies suggest they inherited these blood types from shared ancestors with Homo sapiens. While their Rh factor remains uncertain, it is believed that Neanderthals were predominantly Rh positive, as Rh negative is relatively rare and linked to mutations in modern humans. The compatibility of blood type systems between Neanderthals and modern humans supports the evidence of interbreeding and gene flow between the two species. So, if no other hominids had Rh-negative blood, we can rule out inbreeding with hominids, and we can rule out Homo sapiens having the Anunnaki Rh-neg blood when they created us. Otherwise, the population would be 85% Rh-neg and not just 15%. This leaves us with Anunnaki breeding with us and/or Anunnaki adding it to us for a reason. Let's look at breeding with us first. Most mainstream